My dear students, welcome to Anantapur Arts College Media Center. Today we are going to learn about canal system. The learning objectives of today's lesson are you are going to learn about the definition of canal system, the parts that are present in the canal system, and various types of canal system, and also functions of the canal system. Canal system is the unique system that is found in sponges. Sponges belong to porifera. They are pore-bearing animals. This canal system that is unique in sponges is made up of foldings and perforations. Perforations are nothing but pores, that means holes. This canal system is made up of perforations and foldings. Let us see one by one what are the types of canal systems that are present in different types of sponges. Here you can see canal system is the unique system found in sponges. It is made up of folds and perforations. These folds may be invaginations, that means inward foldings, or avaginations, that means outward foldings. Like that, this canal system, which is unique in sponges, is made up of folds and perforations. The sponge body is organized, that means it is structured to form pores and canals. These pores and canals constitute a unique system called canal system. Alternating invaginations and evaginations result in the formation of certain chambers, of course. Let me explain the different parts of the canal system. Let me make a review of the previous slide. Canal system is the unique system that is found exclusively in sponges. This system is made up of perforations and foldings. And this alternating invaginations and evaginations result in the formation of canal system. This canal system is made up of ostia, which are nothing but the pores through which water enters into the body of the sponge. Spongiocele, which is a cavity that is present inside the body of the sponge. Osculum, this is the exit. That means osculum is the pore through which the water goes outside. Here you have seen two types of pores, namely ostia and osculum. Ostia are the inhalant pores. That means through ostia, the water enters into the body of a sponge. And the cavity that is present inside the body of a sponge is called spongiocele. Whereas osculum is the structure, it is nothing but a big opening through which the water is drained out. Here you can see the typical canal system diagram. I will explain the different parts now. Here, this is called ostia. This part is called ostia. Here, this one, this is called ostia. Ostia are nothing but the openings through which water enters. And this is called this cavity that is present inside the body of a sponge is called spongiocele. And here you can see the upward opening is there. This upward opening through which the water goes out is called osculum. Now you have learned about three parts. One is ostia and another one is spongiocele and another one is osculum. Ostia are the pores through which water enters into the body of a sponge. Spongiocele is the cavity and osculum is the opening through which the water goes out, of course. This forms a simple canal system. And in the subsequent methods, that means in the subsequent models of the canal system, you can find other parts also. That means it is a very simple canal system. Of course, simple canal system contains ostia, spongiocele and osculum. But here in this diagram, you can also see certain foldings, certain evaginations, that means outward foldings, and invaginations, inward foldings. This inward foldings form a cavity, and this canal is called incurrent canal. This canal is called incurrent canal. And this evagination results in outward folding, and this is called, here you can see one more chamber, and this chamber is called radial canal. This is called incurrent canal, and this one is called radial canal. Let me explain. The radial canal and the incurrent canals, they run parallel to each other. Ostia open into incurrent canals. Ostia open into incurrent canals like this. And this is a radial canal, of course. Radial canal, this is radial canal, and this is incurrent canal. Radial canal and incurrent canals, they run parallel to each other. They run parallel to each other. And incurrent canal contains one pore called ostia through which water enters. And whereas this incurrent canal and the radial canal, this is the radial canal and this is the incurrent canal, which are running parallel to each other. These two are communicated through one pore called prosopyle. So what is prosopyle? Prosopyle is the opening through which incurrent canal opens into the radial canal. And the radial canal opens into one more canal called excurrent canal. In this diagram, that excurrent canal is not shown, of course, because of its type, because of its unique type. And excurrent canal will be communicated with radial canal. That means the radial canal and the excurrent canal communicate with each other through another opening called apopyle. And these are different parts of the canal system that are found in different evolutionary models. Just I have listed out the different parts, different integral parts that are found in the canal system. Let me make a review of the same now. Canal system is the unique system that is found exclusively in sponges, and this canal system is made up of perforations and foldings. Foldings may be evaginations or invaginations. And there are three types of canal system, of course. Whatever may be the type of the canal system, there are certain typical structures that are associated with canal system. One is ostia. Ostia are nothing but the pores through which water enter. And another one is a spongocoel. This is the cavity that is found inside the body of a sponge. And another one is osculum. This is another pore through which the water goes out. Apart from that, there is radial canal and incurrent canal. Incurrent canals and radial canals run parallel to each other. Incurrent canals open into radial canals through prosopiles, whereas radial canal open into excurrent canal through apopiles. That means if you just list out, if you make a list of the parts present in different canal systems, you find ostia, incurrent canals, radial canals. Between the two, there is prosopyle. Radial canal opens into excurrent canal through apopyle, and spongocoel is the cavity. Osculum is the outward pore through which the water simply is drained out. These are the parts that are available, that are present in different types of canal system. This is the bird's eye view, of course. Now we are going to classify the canal system and see the parts specific to each model. Let me repeat, canal system is divided into three types, namely Ascon, simplest type. Second one is Sycon, little bit evolved. And the third one is Leucon type, which is greatly complicated. Incurrent canals, I have explained this. Incurrent canals, these are the pores formed by the invaginations of the body wall. That means these are the canals which are formed by the invaginations of the body wall. They contain certain pores, of course, through which they can communicate with the radial canal. Those pores are called prosopiles. These canals communicate to outside through ostia and end blindly at inner side. So incurrent canal, they communicate with outer side through ostia. That means through ostia, the water enters into the incurrent canal. And at the same time, this incurrent canal end blindly at the inner side, of course. But These incurrent canals communicate with radial canals by one opening, and this opening is called prosopyle. So what is prosopyle? Prosopyle is the opening through which the incurrent canal communicate with the radial canal. Then what are radial canals? Radial canals are formed by the evaginations of the body wall. These canals are lined with choanocytes. You know that choanocytes are the unique cells that are found exclusively in sponges. I will show the diagram of choanocytes later at appropriate time. And these choanocytes are nothing but flagellated cells, of course. These are also called collar cells. They make the water currents possible. So like that, here you can see osculum is there, and spongiocele is there, radial canal and incurrent canals are there, and these radial canals and incurrent canals are communicated with each other through one opening called prosopyle. Let us go to the next slide. What are apophyles? These are the openings of the radial canals into the spongiocele, and they can be closed or opened by contractile myocytes. And excurrent canal, these are formed between spongiocele and the radial canal. It is a short, wide canal. This is occupied by flat pinacocyte cells. Coming to the spongiocele, this is the central cavity of the sponge. It directly opens outside through the osculum. And osculum is the opening through which the water is drained out. And here you can see the parts that are represented in this diagram. And I told about collar cells, which are nothing but choanocytes. You can see these choanocytes, which are very unique, and they contain flagella and they also contain collar. Because of the presence of collar, they are called collar cells, and because of the presence of flagellum, they create water currents inside the spongiocele. Normally, these choanocytes are present in the endoderm in simple sponge. Hence, the endoderm of any sponge is called choanoderm. Whereas in case of complicated sponges which contain complicated canal systems, these choanocytes may be confined to the radial canals, and in some more complicated sponges, they may be confined only to the flagellated chambers. And let me explain about the types of canal system, namely Ascon type, Sycon type, here you can see, and one more thing, Leucon type. Ascon type is the simplest canal system. Sycon type is a little bit complicated, and the Leucon type is highly complicated. Here you can see the diagrammatic representation. You see this Ascon type contains ostia through which the water enters, and this cavity is called spongiocele, and you can find the opening, big opening at the top of the sponge. This is called osculum. That means in Ascon type of canal system, you find only ostia, spongiocele, and osculum. Ascon type, it is the simplest canal system. Example is Leucoselenia and Oleanthus. Oleanthus is nothing but undifferentiated stage of sponge. The body surface has ostia. Ostia open into spongiocele, which opens outside through a narrow circular opening called osculum. Osculum is bordered by large monoxen spicules. The water flow is maintained by beating of flagella and choanocytes. To sum up, my dear students, this Ascon type is the simplest type, which contains ostia, spongiocele, and osculum. That's it. And you can see the water flow, how it follows in Ascon type. Incoming water enter into ostia, and from ostia they enter into spongiocele, water enter into spongiocele, and it goes outside through osculum, of course. Through osculum, it is going out. And this middle diagram represents Sycon type. It is little bit complicated when compared to Ascon type. Here you can see in Sycon type, the margins, that means the layers are showing certain invaginations and evaginations. Whereas in Ascon type, such invaginations and evaginations are not found. In Ascon type, there is only ostia, spongiocele, and osculum, whereas here you see the body wall, how it is showing invaginations and evaginations. So, in these invaginations and evaginations, the ostia are opening into invaginations, which form incurrent canals. Incurrent canals are formed by invaginations and they communicate with outside through ostia. That means ostia open into incurrent canals, and later here you can see inside, that means because of the evaginations certain chambers, inner chambers are formed. These are called radial canals. Like that, these incurrent canals and radial canals, they run parallel to each other, and here you can find one inner chamber, and this inner chamber is called spongiocele, and here you can also find osculum through which water is going out. That means in case of Sycon type of canal system, you can find ostia which open into incurrent canals, and radial canals are also there, and spongiocele is there, and spongiocele opens to outside through osculum. This is about another type, namely Sycon type. And let us see another type, Leucon type. Let us go back to the diagram. Of course, here you can see the Leucon type. You can see the basic difference between Leucon type and other types of canal systems. In Ascon type and Sycon type, you can find a cavity called spongiocele. In Ascon type, you can see ostia, spongiocele, and osculum. In Sycon type, you can find some extra structures, namely along with ostia, you can also find incurrent canals, radial canals, spongiocele, and osculum. Whereas here you observe specifically spongiocele is absent in Leucon type. In the Leucon type, spongiocele is totally absent, and it is complicated, and it shows extra foldings forming flagellated chambers. That means here one more additional structure has crept in, and this is called flagellated chambers. We'll make it very clear by describing the entire nature of the Leucon type in the next slide. Here you can see it is very complex. Leucon type is very complex. Ostia enter into incurrent canals. Incurrent canals and radial canals are communicated through certain pores called prosopiles, and radial canals open into excurrent canals. These excurrent canals and radial canals communicate with each other through one more opening called apopiles. If excurrent canal is absent, the apopiles may communicate directly with spongiocele, of course, but here in case of Leucon type, because spongiocele is absent, the apopiles, they establish a communication between excurrent canals and radial canals. So that is about apopiles and prosopiles. Let me repeat. Prosopiles are the structures that establish communication between incurrent canals and radial canals. Apopiles communicate the radial canals with excurrent canals. That means the pores present between radial canals and excurrent canals are called apopiles, and the pores present between incurrent canals and radial canals are called apopiles. So here you can see the diagram. You can see in this diagram, you can see very clearly there are certain chambers called flagellated chambers. In Sycon type, we have used one term called radial canals, but in addition to that, there are certain chambers called flagellated chambers in Leucon type. So like that, Leucon type contains ostia, incurrent canals, flagellated chambers, excurrent canals, and holes or pores or perforations like prosopiles, apopiles, and osculum. And you can see the spongiocele is not there. That means it has vanished. It is obliterated with various invaginations and evaginations like flagellated chambers. Let me read out this. The ostia open into the incurrent canals, which open into flagellated chambers through prosopiles. These chambers open into excurrent canals through apopiles. So here the connection between incurrent canal and flagellated chamber is prosopile. Like that, the connection between these flagellated chambers and excurrent canals are nothing but apopiles. So that is about the complication of the Leucon type. And coming to Ragon type, of course, in Leucon type, there are 3 more evolutionary models. We will come to that in the next slide. Let us see this Ragon type. This Ragon type is not found in any adult sponge, but it is found in the larval stages. It's simply triangular and pyramidal in nature, either triangular or pyramidal in nature. And let us see the different models of the Leucon type. In Leucon type, I told that spongiocele is totally absent, and you can find many new chambers called flagellated chambers along with ostia, incurrent canals, flagellated chambers, excurrent canal, osculum, apopiles, and prosopiles, etc. Based on the presence of these parts, Leucon type is differentiated into uripilus type. Here you can see the flagellated chambers open into excurrent canal through broad apopiles. That means in uripilus type, the perforations called apopiles are present. And coming to apodal type, in this type, flagellated chambers open into excurrent canals through narrow canal, not narrow pore, narrow canal, and this canal is called apodus. Apopile is the pore, apopile is the hole, apopile is the perforation, whereas apodus is nothing but a narrow canal. I hope you understood the difference. Apopile is the pore, whereas apodus is nothing but the narrow channel. And diplodal type contains the incurrent canal where it opens into flagellated chambers through narrow canal called prosodus. If prosodus is present, it is called diplodal type, and if only apodus is present, it is called apodal type, and if apodus is absent and apopile is present, it is called uripilus type. That means Leucon type is divided or classified into uripilus, apodal, and diplodal type. In uripilus type, apopiles are present. In apodal type, a narrow canal called apodus is present. In diplodal type, what is happening? Prosodus is also added. Prosodus is a narrow canal that connects between or that connects the two structures, namely incurrent canal and the flagellated chambers. Here you can find the diagrams of apodal and the diplodal. So that is about the different types of canal system. Let us make a summary within few seconds. Canal system is the most unique system that is found exclusively in sponges. Canal system is divided into Ascon type, Sycon type, and Leucon type. Ascon type is the simplest system which contains ostia, spongiocele, and osculum. Whereas the Sycon type is little bit complicated, that contains certain structures, additional structures, along with normal structures. This Sycon type contains ostia, incurrent canals, radial canals, spongiocele, and osculum. And spongiocele is present both in Ascon type and Sycon type. Coming to the Leucon type, spongiocele disappears and spongiocele is not found. There are many complications. There are many invaginations and evaginations. And in Leucon type, you find certain structures called ostia, incurrent canals, prosopile, flagellated chambers, excurrent canal, apopiles, and osculum. Like that, different complicated structures are found in Leucon type, and it is divided again into uripilus, apodal, and this diplodal type. So like that, there are different modifications and evolutions of the canal system model. And whatever may be the model of canal system, whatever may be the type of canal system, the functions are nutrition, circulation, excretion, and reproduction. So nutrition is accomplished by canal system, followed by circulation, excretion, and reproduction. So my dear student, it is a stock question for many examinations in the zoology stream. Try to learn it meticulously. Thank you very much. Meet you with some other lesson at some other time. Thank you very much.